In a previous video, I talked about the structure of cytoskeletal elements. I talked about microtubules in depth, but I also introduced microfilaments and intermediate filaments. In this video, I'm going to go a little bit more in depth into the structure of microfilaments or actin filaments, and I'll also talk about the proteins that are associated with actin filaments, particularly myosins, which are motor proteins that walk along actin filaments. As you already know, microfilaments are made from two strands of actin monomers that form polymers, and those two strands are wound together. We call the actin monomers G-actin. So we have these G-actin subunits that are the monomers. The G-actin subunits form these polymers that we call F-actin, and two F-actin polymers are wound together to form our microfilaments. Just like microtubules, microfilaments have what we call a plus end and a minus end. And just like in microtubules, the plus and the minus doesn't refer to charges on the structures. It refers to the growth of those structures. Microtubules only grow on the plus end, but microfilaments grow on both the plus end and the minus end. They just grow faster on the plus end than they do on the minus end. In microfilaments, we add the G-actin monomers when they are bound to ATP. If you recall, in microtubules, tubulin dimers were added to the microtubules when they were bound to GTP. So microtubules would add GTP tubulin subunits. In actin filaments or microfilaments, we add ATP G-actin. Just like in microtubules, when the GTP was hydrolyzed to GDP, those monomers or the tubulin dimers would fall off the microtubules. In microfilaments, when the ATP G-actin is hydrolyzed to form ADP G-actin, those monomers are going to fall off the microfilaments. And generally, the rate of addition of our actin monomers is dependent on the concentration of free ATP G-actin. So the more available ATP G-actin there is, the faster they will add to our microfilament. After an ATP G-actin joins the polymer, the ATP is hydrolyzed to ADP. That ADP G-actin will quickly dissociate if it's at the end of the filament. Again, this is very similar to what we saw in our microtubules. So therefore, if a new ATP G-actin is added before the last ATP G-actin is hydrolyzed, the polymer will grow. But if the last ATP G-actin is converted to an ADP G-actin before the next ATP G-actin is added, the polymer is going to shrink. So we have this race with the microfilament where we're trying to add new monomers before the previous monomers are converted to ADP G-actins. If we can add new monomers before that hydrolyzation occurs, the filament will grow. If we can't, the filament will shrink. So again, like microtubules, the actin monomers will grow if the concentration of the ATP G-actin subunits is high. So if we have enough ATP G-actin, we'll grow our microfilaments. But unlike microtubules, in microfilaments, we'll add to both ends. So let's move our positive and negative sign here on our microfilament to give us some room. We have the plus end over here. This is the minus end, and again what that means is we're going to grow faster on the plus end than we will on the minus end. So we can see we're adding those ATP G-actin monomers. We added more on the plus end than we did on the minus end. As we're adding those monomers, the concentration of ATP actin that's available to be added is going to decrease. We're taking those from the cytoplasm, we're adding them to our microfilament, so the concentration of the free ATP G-actin is going to decrease. That means we're going to reach a point where the G-actin is still being added to the plus end, because remember, they're added faster on the plus end than they are on the minus end, but they're being added at the plus end faster, they're being added at the plus end at the same rate as they're being lost from the minus end. So we'll get to a point where we'll lose these ATP monomers from the minus end, but we'll still be adding them to the plus end. So now our polymer is at steady state. it's not growing or shrinking. It's losing monomers from one end at the same rate as it's adding monomers to the other end. What this means, though, is that the relative positions of subunits is going to be moving even though the polymer isn't growing. If you just look at this specific monomer, for example, as we are removing monomers from one end and adding monomers from the other end, this monomer that we are tracking is going to move relative to the ends of the filament. In the top image here, it is right about the middle of the filament. In the bottom image, it is near the end of the filament. It's near the minus end. So it's moving relative to the plus and minus end of the filament, even though the polymer isn't growing. And we call this process treadmilling because it looks like a treadmill, right? The length of a treadmill doesn't change as it moves, but the position of objects on the treadmill do change as the treadmill moves. The concentration of ATP-G-actin at the steady state is what we call the critical concentration. So if the concentration of ATP-G-actin is higher than the critical concentration, the filament is going to grow. If it's lower than the critical concentration, the filament is going to shrink. And if it's at the critical concentration, you're going to see this steady state process of treadmilling. I'm now going to transition and talk about the proteins that are associated with microfilaments. Most of the rest of the video I'll be talking about myosin, the motor protein that walks along actin filaments, and I will talk about a specific example of myosins acting as motor proteins, which is muscle contraction. Myosins are a very diverse group of motor proteins. There's about 20 different classes of myosins in eukaryotic cells. All of the myosins are going to convert energy from ATP hydrolysis into mechanical work. They're going to walk along actin filaments by hydrolyzing ATP or using the energy that is released by hydrolyzing ATP. Humans alone have about 40 different genes for myosin that are from 12 different classes of myosin proteins. And all the myosins are going to move along actin, but they'll do different jobs. They'll carry different cargo. What we call conventional myosin, or myosin 2, is the myosin that was found in muscle, and it's the best understood. We call the unconventional myosins all the other myosins, even though there's more of them. They're unconventional because they weren't discovered, or they haven't been as well studied as the myosins that are involved in muscle contraction. But all of the myosins, all the conventional myosins are plus N directed, and all but one class of myosins, the unconventional myosins, are also plus N directed. So the myosins are almost always going to be walking towards the plus end of the microfilaments. All of the myosins have a head that binds actin and hydrolyzes ATP, and that's what you're seeing in the schematic here. You're seeing these myosin heads that are walking along actin filaments. The heads are bound to the actin. The heads is where ADP and ATP are bound. ATP is hydrolyzed. And then we have tails where you're going to have the cargo attached. Now let's take a little bit closer look at the structure of myosin 2, which is the myosin that's involved in muscle contraction. Humans have 16 different myosin 2 heavy chain genes. And the key point of this is not the exact number of genes, but the key point is that even though I'm going to give an example of muscle contraction and talk about myosin as if it's one kind of molecule walking along actins, even the head piece of myosins in humans are coded for by a bunch of different genes. There's lots of variability in myosin motor proteins. The myosin structure is such that, again, they are plus N directed motor proteins for the most part. The myosin 2s are all plus N directed. And they have 6 polypeptides, so they are a protein made from 6 polypeptide chains. They have these globular heads, and you can see there are 2 of those head pieces. So we have this globular head, and there's a tail, and that tail is composed of 2 long alpha helices. And so we have these 2 alpha helices that are wound together, and the tails in myosin 2 are going to form these long filaments, and these myosin filaments are due to those myosin 2 tails that are all joined together. These filaments are what we call bipolar. there's clusters of myosin heads at both ends of the filament, and that will look or be a little bit more clear in a moment when we look at the structure of muscle cells. I'm going to use skeletal muscles to demonstrate how the myosin motor proteins walk along actin filaments. But to do that, we need to understand the structure of skeletal muscles a little bit. So skeletal muscle cells, or muscle fibers, are large and cylindrical cells. If you look here at this major structure, we have the bone. The bone has a tendon that's attaching it to our skeletal muscle. The skeletal muscle is divided up into subsections, and within those subsections there are muscle fibers. Those muscle fibers are our individual cells, and as you can see from this diagram, they are these long cylindrical cells. They are roughly 100 millimeters long. There's lots of variability, but they're several dozens to 100 millimeters long, and they are 0.01 to 0.1 millimeters wide. You can compare this to a typical mammalian cell, which a normal cell would be 10 micrometers in diameter, and so rather than being 10 micrometers in diameter, these are 100 millimeters long. These cells are so big because they are multinucleated. There's up to 100 nuclei in each of these muscle fibers, and they become multinucleated by going through the process of mitosis, but they don't go through cytokinesis. So the chromosomes double, they create more nuclei, but they don't then pinch off the cytoplasm to create unique cells. So we have these multinucleated cells that form the muscle fibers. Each one of the muscle cells has many myofibrils in it. Those myofibrils are going to be composed of many sarcomeres. So you can see highlighted on this image one of the sarcomeres, but each one of these dark blue lines to dark blue lines is a sarcomere. And so we have many of these sarcomeres that are lined up to form a myofibril. A bunch of myofibrils bound together are going to form a muscle fiber. Let's now take a look at the structure of that sarcomere. The sarcomere is about 2 micrometers long at rest, and during the process of muscle contraction, they're going to shorten up to 70%. So you can envision if we have this muscle, this myofibril here, if each one of these sarcomeres shortens up to 70%, you're going to have a dramatic shortening of that entire muscle. So an individual sarcomere only shortens a little bit, right? It's only 2 micrometers long, so when it shortens, it's only shortening by a micrometer or so. But if you add a whole bunch of those together, you can get a lot of distance in contraction. Those sarcomeres are composed of actin filaments. We call those the thin filaments. So here we have actin filaments, and they're composed of the myosin chains, which we call the thick filaments. A couple moments ago, when I was talking about the structure of those myosin II filaments, I talked about the idea that they were bipolar. They had myosin heads on both ends. So we see myosin heads here, and we see myosin heads here, and those tails are twisted together to form that filament. I also mentioned that those myosin motor proteins were plus end directed. So they're going to walk along our actin filaments towards the plus end. So these myosin heads are going to walk in this direction along these microfilaments. They're going to walk in this direction along these microfilaments. And each one of these myosin filaments is composed of hundreds of myosin proteins. The plus end of the actin then is anchored at what we call the Z disc or the Z line. I've heard both terms. In fact, I think your textbook uses both terms. And so the Z disc is where we have the plus end of the actin anchored, and as you can see from the schematic, here is the next sarcomere that's starting. So here's the plus end of an actin filament, and if we were to extend this way, you would see then another set of myosin filaments on this end on the next sarcomere. I'm going to use this schematic diagram of the actin and myosin filaments in order to demonstrate how they slide relative to each other during muscle contraction, and then we'll look at how the myosin heads actually walk along the actin filaments. So just like you saw in the previous slide, we have the Z line, which is the middle of the thin filament. So here's Z line of one sarcomere, Z line of another sarcomere. Here's another Z line that's unlabeled because I'm about to add some more text under here. But here's the other Z line. So each one of these Z line to Z lines is a sarcomere, and the actin, the thin filaments are composed of actin. One actin filament will have its plus N anchored at the Z line. Another actin filament will have its plus N anchored here at the Z line. The M line is the middle of the myosin, so we have an M line here, an M line here, an M line here. We call the I band the region of actin that is not overlapping with myosin. So this section of the thin filaments here is our I band. We have an H zone. The H zone is where there is no actin, so we have myosin only. And then the A band is where we have the whole length of that thick filament. So the A band is the width, the length of the myosin. During contraction, our Z lines are going to move inward. So this Z line will be moving in this direction to the left. This Z line will be moving to the left. On the other side, this Z line will be moving to the right. This Z line will be moving to the right. The H zone is going to shrink. This region where there is only myosin and no actin will shrink. The I band will shrink, the region where there is actin and no myosin. But the length of the myosin isn't going to change. And this is a key part of what's happening. The length of our filaments don't change, but the filaments are going to move relative to one another. So let's see how that looks. When the muscle contracts, you saw the I band and the H zone shrink, but the A band didn't change. So let's look at that again. We see the Z lines moving inward, and we see the H zone and the I band shrink, but the A band stays the same width. This is called the sliding filament model of muscle contraction. Again, the filaments don't change shape. The actin and the myosin are staying the same length, but those filaments are going to be moving relative to one another. Let's look at now what's happening to the myosin and the actin during this contraction. So this is the model for the actomyosin contractile cycle. In muscle contraction, it begins when calcium binds to the protein troponin. Troponin is covering the active sites on the actin where the myosin binds. So calcium is going to signal for the troponin to be moved away from the binding site and allow the myosin to interact with the actin. And you can see this little blue dot here is indicating where on the actin filament this myosin protein is going to attach. Initially, a myosin head that is bound to ADP and a phosphate group is going to bind to the actin. The binding to the actin is going to cause the ADP and the phosphate to be released, and that's going to cause the myosin to bend and move the actin filament. So as that myosin bends, the actin filament is going to slide to the left. We call this the power stroke. The binding of ATP to the myosin is going to cause the myosin to release from that actin filament and then straighten out again. And then the hydrolysis of the ATP is going to cause the myosin to change to what we call the cocked conformation. So the position where it's ready to bind again to the actin for another cycle. As long as there is enough calcium in the muscle cell to keep the active sites on the actin exposed, then we're going to repeat steps 2 through 5, and the myosin will again bind to the actin. It will bend to move the actin. It will release, and then it will attach again and keep going through the cycle, again, as long as there's enough calcium to allow the active site to be exposed. And as you can see from this diagram, the myosin is walking towards the plus end of the microfilament. Right here we have where the site where the myosin is binding. The myosin bends and slides that in the negative direction, and so the myosin is going to bind again further down our microfilament. So the myosin is walking towards the plus end of that actin filament. Myosin is a protein that binds to actin, but there are a bunch of other actin binding proteins. I'm just going to use this diagram on the right of a neural growth cone to talk about just a subset of actin binding proteins. So what you're looking at here on the right is a growth cone in a Drosophila neuron. So neurons have these axons. These axons are going to make connections with other cells. They need to grow out from the cell body, and they grow by extending these filopodia, and they reach out, and when they make contact with a signal molecule, the axon will follow those filopodia. These filopodia are extensions of the cell membrane that are caused by actin filaments. In the schematic here on the left, you can see those actin filaments that are pushing out those filopodia. The red here is representing actin filaments. The blue is representing microtubules. In the previous video, I talked about the idea that the axons have microtubules in them that ship proteins to the end of the axons. What you're seeing up here in these nice fluorescent images is the microtubules labeled in green and the actin filaments labeled in purple. What I'm going to do is just zoom in on this portion of the diagram. This panel D is a schematic of just one of these filopodia here, and it's highlighting a bunch of different functions of actin binding proteins. So again, what I'm about to do is not talk about all the different kinds of actin binding proteins that are out there, but the next slide is intended to give you an idea of how many different proteins are associated with actins and how those proteins can result in different functions and different properties of actin filaments. We'll start with these proteins that are labeled number 1. These are monomer nucleating proteins. The actin filaments, as I've talked about in this video, grow primarily on the plus end. So these monomer nucleating proteins you see are identified here on the minus end of those microfilaments. And the idea is that to get this process started, we need to have a couple actin monomers come together, and these monomer nucleating proteins promote the nucleation of those actin filaments. They get the process started. The proteins that are represented by 2 and 3 are monomer polymerizing proteins. They're going to facilitate or catalyze the addition of those G-actin monomers, and they're going to help those microfilaments grow. We also have end blocking or end capping proteins. That's represented by this number 4 here. These end blocking proteins are going to regulate the length of the filament either by preventing polymerization, so they're going to prevent the addition of even more actin monomers, or they might be preventing the depolymerization, so they might stabilize those actin filaments so they can't shrink. What you see represented by the number 5 here with these scissors are what we call filament severing proteins. As I've talked about in this video, ATP G-actin monomers are added to filaments, but ADP G-actin monomers fall off filaments. So if you want to break up a microfilament quickly, what you need to do is expose the ADP G-actin monomers, which are in the middle of the filament. Right? The ends have the ATP G-actin monomers, but once we get to the middle, we are looking at largely ADP G-actin monomers. So if you cut off the end of one of these microfilaments, you're going to expose ADP G-actin monomers, which is going to facilitate the breakdown of those microfilaments. What you see here represented by number 6 are cross-linking or bundling proteins, and these are going to create three-dimensional microfilament structures. So we can bundle microfilaments together in different orientations to create larger structures inside the cell. There are proteins that are involved in the movement of microfilaments. What you're seeing represented by the number 7 are retrograde flow proteins. So proteins that are going to move the actin cytoskeleton away from the end of the cell, so in this case away from the filopodia back towards the cell soma, and these are going to move actin filaments, and so it's a way to quickly remove those actin structures from an area. It can be faster than depolymerizing those actin filaments, and so we have proteins that will move entire filaments. And lastly, proteins 8 and 10, and this and number 9 isn't shown on this slide. Number 9 was in panel C of this figure that was on the previous slide. But 8, 9, and 10 are different examples of microtubule binding proteins. And these microtubule binding proteins are going to allow actin to interact in different ways with the microtubules. I've been talking about microtubules in the last video and then actin filaments in this video, and I largely have talked about them as separate cytoskeletal components, but all the different cytoskeletal elements are going to interact with each other. And so we have lots of interactions between microtubules and cytoskeletal elements. They don't act or exist in isolation. They're going to work together. Between this video now and the last video, I've talked a lot about the specifics of the structures and the associated proteins of microtubules and microfilaments in particular. There's lots of similarities between the structures of microtubules and microfilaments. They're polymers made from monomers, for example. They have motor proteins that walk along them. But it's important to keep in mind that these filaments, microfilaments and microtubules and intermediate filaments, all the different cytoskeletal elements, work together, and that a lot of the functions of those cytoskeletal elements are due to the other proteins in the cells that are going to interact with those cytoskeletal structures. So even though we talk about microfilaments playing a role in locomotion or movement of muscle cells, for example, we talk about microtubules playing a role in the movement of cells such as cilia and flagella. What is allowing those microfilaments and microtubules to do the things they do in the cells is typically the proteins that are associated with those microfilaments and microtubules. The microfilaments and the microtubules provide the structure, and the other proteins provide all the activity that is facilitated by those cytoskeletal elements.